want to start with a big thank you to Allison Pierce with Audubon Naturalist Society for inviting me to talk with you today and for recommending today's topic about bioblitzing, or doing this backyard survey. I teach wildlife courses at University of Maryland, such as wildlife management techniques, which involve surveying wildlife and their habitat, so this topic is already exciting to me. Plus, with tonight's talk, I get to add in this unique spin to my day job by focusing this talk using some tools you may already have around your home. I also want to thank Audubon Naturalist for hosting the talk, Serenella for testing the online hosting with me, and of course, I want to thank all of you who registered, who logged in, and who are going to be participating. I'll do my best to combine several semesters worth of classes into 1 hour. All right, so what is a bioblitz? So this is an event where someone inventories as many living species as possible at a given location in a given time frame. Most common is a full 24-hour bioblitz with multiple teams to cover the full 24 hours. But you get to decide the details about where, when, how, and what will be surveyed. It can be done by one person or more. It can be done on one day or night or more. In the year of do-it-yourself everything, every day, staying at home every day, I thought, let's do something good with our time. With technology allowing us to access new places and share data, we're identifying new species all the time. Here are the current number of species broken down by taxa. These numbers are in the thousands unless noted by millions. And these numbers are staggering. Look alone at the number of insects. There's over 5 million species, that's species, not individuals, in unique species of insects. How many do you think are in your backyard? How many species of mosquitoes alone do you think are in your backyard? You may have heard of things like Asian tiger mosquito, yellow fever, common house mosquito, but they're up to 60 species living in Maryland. And how are you going to know how many are in your backyard until you do a bioblitz? Do you want to know the total number of all living species in your area, or the total number of animal species, or just the number of bird species in your area? You'll want to decide this in advance. You'll also want to consider scale. So let's start with timing. Are you interested in knowing the number of species in one given time frame, like the number of permanent residents or winter residents, or the number of spring migrants or fall migrants? Or do you want to know the number of species year-round? This will also help you decide how you're going to run your bioblitz. You also want to think geographically. Are you interested in learning the number of species in your backyard, in your whole neighborhood, or at a local park, perhaps? Typically, the larger the area you're interested in surveying, the more volunteers you're going to need. And many of those volunteers will feel some ownership in that area. And working on the bioblitz together can lead to deeper connections between you and them and the ecosystem. Some other considerations before deciding on your question are the tools you can access. If you don't have all the tools you need, do you want to narrow the scope of your question based on the tools you already have? Or do you want to purchase additional tools? Or do you want to borrow some tools after disinfecting for COVID, of course? Do you want to keep your question broad or narrow it down? If you want to keep it broad and you don't have all the tools, do you want to ask a friend or a neighbor who has the expertise and the tools to join you during your bioblitz? Another thing to consider is the data and how you're going to report it. Are the results just for you, or are you wanting to help the scientific community in general? Whether or not you want to keep the data private or share it, I still recommend documenting your species using iNaturalist, because it can serve as a centralized database for you and can help contribute to science if you want to open up the permission. And selfishly, I hope you open up this findings to everyone so that I can see where the hotspots are if I'm looking for a certain species. Here's a list of things you can do to increase the number of species you see during your BioBlitz. Remember that some species are nocturnal, so you'll need to be as well if you want to have a better understanding of the species that are on your landscape. And surveying more than one day can yield more species since certain individuals may have been avoiding your landscape for a variety of reasons. Maybe they were avoiding predation, avoiding competition, avoiding your new BioBlitz survey activity. It can take several days or weeks for wildlife to acclimate to change. So don't be surprised if you conduct surveys every day and it's not until day 3 or 4 when you start seeing new species. Also, remember that different species thrive in different areas, so you want to survey different kinds of habitats to make sure that you're surveying across your whole landscape. For example, you'll find different species at different depths of pond, towards the shore, on the shore, far from the shore, under the leaf litter, in the understory, mid-story, or in the canopy. And I can't stress enough the documentation. So documenting as much data as you can, collecting quality data. So get quality recordings, pictures, whether the recordings are sound or video. You can use more than one observer, and this helps many times with additional observation. You can also use iNaturalist to help you with your identification as well. So that's another reason to use iNaturalist. Let's start with common survey techniques used for mammals. So I'm going to talk about camera trapping, owl pellet surveying, reviewing skulls, reviewing tracks, and also looking for scat. Camera traps started off expensive, but are becoming more affordable. And do-it-yourself Wi-Fi home security cameras are becoming very affordable. Most of them haven't been waterproof in the past, but this summer, Wise is going to come up with their first weatherproof, cordless, cloud storage, outdoor security camera. This is going to revolutionize the camera industry and make surveying for home security, but really I use it for wildlife species. Regardless of the type or brand of camera that you're using, you get to set the sensitivity to movement or the trigger speed. So the time it takes from when there is movement to when it takes the first picture or video. And you can preset time intervals. You can have it take a picture or video once an hour or several times an hour or continuously once it's triggered. SD cards and cloud storage have become more affordable recently, meaning you can take higher quality resolution pictures, and many of them. You can set that sweet spot or the number of pictures per event and adjust that balance accordingly. But with SD cards being more affordable, you can usually afford larger space to hold your data, so you can usually afford more pictures and higher resolution. So thinking about where to set up your camera is extremely important. You can use a tree that's been used typically for these kind of surveys, or you may have certain kinds of posts in your yard that may work as well. But the goal is to think about the location, the kind of bark or the substrate you'd be mounting your camera to, the diameter of what you're mounting it to, and the height. All of those are going to influence the data you're going to gather or the species that you're going to detect. So as you're thinking about location, you want to put it in an area you believe wildlife exists. And when you're thinking about location, you want to think about the bark and the DBH. You want to try to pick a tree species that has rough bark, not smooth bark, so the camera doesn't slide up or down, so it stays put. And you want to pick a diameter of your tree or whatever kind of pole you're mounting to. You want to pick a diameter that's greater than the width of your camera. Again, so when you secure it, the camera doesn't slide up or down. You want the height of the tree or pole to be tall enough so that someone doesn't come and slip the whole setup over. You want to find an appropriate camera angle that's parallel with the ground. You don't want it aiming too high where you're aiming towards the sky. Therefore, you're missing the height of your animals. And you don't want to aim it too low towards the ground where you're missing the top of your animals as they go over that camera. So you want to make it parallel to the ground. And you want to adjust that height. Usually for larger mammals, so deer, let's say white-tailed deer, we usually place the camera around hip height. For meso-sized carnivores, we usually place our camera around knee height. And for small mammals, we usually place it around ankle height. We want to secure it to a tree at that height once we're pretty confident that that is a good location and a good angle. And we'll know it's a good location and angle when we do this walk test. So we can set it in this walk test, like a test mode, where we can stand back from the camera and keep stepping back and stepping back to see if the light goes off. That tells us that we triggered the camera as if it were going to take pictures. So it's still detecting us. We want to aim for about 30 meters or farther of a detection range where that camera still sees us and still takes pictures. Because if it stops detecting us at, say, 5 meters, then that means it's only going to detect animals that were very close to the camera, and it won't see animals that are farther away. And so you may need to readjust the angle or the height of your camera. Once you're pleased with your walk test, then you can arm the camera or set it to turn on so that when it's triggered, it'll start taking pictures. Then the next day, or whenever your BioBlitz is done, then you go and download your pics and have fun identifying them and uploading them to iNaturalist. So that's it for the camera trapping. Are there any questions at this point?
We do have one question, our first question, actually, asking if you could speak about the type of cameras to be used.
I have a lot of experience with Reconyx. Specifically, I like Reconyx Hyperfire. There's like a 500 series and an 800 series. The price has been a little pricey, maybe $400, $500 or so. I like the resolution. I like the ease of setting everything up. I like all the options I can pick from. So the trigger speed, the resolution, quality. I like all that. So I really like Reconyx. I've tried a few other brands and found that the light was really bright and it startled animals away. So I only had that one chance to get a picture before, and they never returned to that area. It startled all the animals away. So I've tried some other brands that I didn't like quite as much as that Reconyx. Just I like that kind of glow, the infrared glow, instead of a bright white light. That's a really stark contrast to the dark night. Also, what I like about most of these brands is during the day you get color photos, and then at night it's relying on infrared, and so it's black and white at night. There's not much we can do about that. But I think I mentioned earlier about Wise. So their cameras are affordable, like $30 or so. So super affordable. And yes, it's meant for home security, but if you change the angle, the position, the location, I use mine all the time for wildlife surveys. And they work really well, and they're super affordable. They're lightweight, almost no one notices them. So I really enjoy Wise cameras, but I'm really looking forward to their weatherproof camera and cordless. So then I can put it all over instead of just being reliant on being near a power source. Okay, are we good, Rob?
Yeah, you're good.
All right, now we're gonna get to owl pellets quickly. So if you're lucky enough to have an owl species roosting on your property, whether it's in a tree or in a barn, you'll notice these pellets under that roost. So these pellets are, they're composed of the non-consumable parts of their diet, and they get regurgitated. They kind of look like these furry little rocks, if you will. And you can open them up with your fingers, and you can use some other tools as well. You can open them up to find these different bone fragments and skulls of what they've ingested. Now, identifying their diet is what we're really after to help us determine what species are in our area. So this requires us learning how to identify skulls and bones. So Maryland DNR has a really great key to learning how to ID species based on their skulls. And we may find skulls and bones in our yard normally. We don't have to wait just for owl pellets. We may walk around and see these quite often on the landscape because the bone lasts a long time in the environment. So we tend to see that much longer after we've seen the cadavers. So we can, it definitely behooves us to learn how to identify skulls, so different parts of the skulls. And it starts by looking at the overall size and form of the skull. We can take a look overall and notice the size and position of the eyes. So we usually see larger eyes in comparison to the skull in nocturnal animals, since they need to let more light in. And we see smaller eyes in our diurnal animals, and then an absence of eyes in our underground burrowing animals. When we look at the position of the eyes in comparison to the skull, we notice the position of the eyes is forward to create this binocular or stereoscopic vision for assessing depth. So we see that often in predators. Then we see the eyes moving more towards the side of the skull and above when we see prey species, so they can see predators coming from all sizes. We tend to see longer rostrums, or the muzzle, if you will, in species that are relying more on smell. So we see these a lot in the canid species. But the key here is really helpful. So I definitely recommend checking out that key. So now moving on to tracks. Most species leave tracks. They're identifiable down to the species level. So you can look for the tracks in their natural environment, such as in snow or on sand or mud, or you can create an environment where they'll leave tracks, such as track tubes or track stations. The idea behind track tubes and scent stations is that you're creating a location where you're baiting the animals into the area. So there's some kind of smell drawing them in, and in turn, they leave evidence of their presence by leaving their tracks. If we place bait in the middle of a PVC pipe or downspout or whatever kind of piping you may have, if you leave bait in the middle of it, you're encouraging the animals to walk in from either side towards the middle of the pipe. This gives you lots of opportunity to create ink pads that they would step on and then transfer the ink that's been touching the surface of their pads that transfers onto another surface. So if we create this ink pad, so basically I take felt, I pour a mixture of carbon black and mineral oil. I'll mix that up. Or Maryland DNR says you can use food coloring and cornstarch. Mix that up. Then you lay that out on felt. So they step on the felt first as they enter, and then they step on whether you want to leave contact paper or if you want to leave white paper. They would step on that. Then they would approach the bait and either eat the bait or eat some and leave through the other side. So this is a great way to see presence of small mammals. And then up at the top right-hand corner, we have a picture of this scent station. It's a very similar concept that we're drawing these animals in to an area, so we're encouraging them to step in a certain area. But what we've done in advance here is we've cleared the area of vegetation, say a 1 meter circumference, or maybe a 1 meter by 1 meter square around a center. And in the center, we place some kind of smelly bait. So we've cleared the area of vegetation, we've leveled it, and then we may lay some kind of fine sifted flour or sand, something when they step on, we'll see the track pattern. So as they step onto it to get to this smelly thing, I tend to use sardines, I've used cat food, I've used tuna, other people have used smelly cheeses. So whatever you think has a lot of smell, and we'll draw them in. It's still a very similar concept of we're creating this artificial area where we're encouraging the animals to step. And then, so we don't touch the animal, but we are going to see evidence of their species, the presence of a certain species. We could couple that with a camera. So we could draw them in and add a camera right there as well. So that's another possibility. But the idea is we can identify species through their tracks, or through their skulls, or through their pictures on our cameras, and we'll keep going through some other techniques. So for tracks, the way we ID them is looking at the track, looking at the front foot and the hind foot, looking at the number of toes on each foot, looking at the number of lobes on the heel pad. And this example, figure B, shows 2 lobes on the heel pad, whereas figure C shows 1. Also, we look for the presence or absence of claws. So in figure B, we don't see claws. In figure C, we do. So figure B, if you go through the key provided by Maryland DNR, you'll see that figure B is a feline species, feline C is a canid species. So felines, for those of us who may have cats as pets, we know they have those retractable claws. So they tend, as they're stepping, they tend to keep them retracted. So we don't see evidence of the claws in their tracks. And then this picture I provided in the top right-hand corner is an example of a feline species, a bobcat. So again, we see those 2 lobes, no evidence of claws, so we would see that this is a feline species, specifically bobcat. We also look to see, is there webbing between the digits? So that's something else that we're looking for. We also are looking at distance between our steps, the width of the gait, and the pattern. But it all starts with knowing which is the hind foot and which is the front foot. So how do we know that? Well, first we have to know which kind of gait or which kind of pattern they use as they walk. So we have our diagonal walkers. So these tend to be our cats and dogs and hoofed animals. So they're moving their hind limbs, so their opposite limbs together. So the right front forefoot with the left back leg. Then we see these pacers. So these tend to be our wider bodied animals. So they're shuffling along. We see bounders. So this is where they're hopping in a series, and as they're landing, we see those hind legs pull up right behind the front legs. So they're not quite on, so the hind legs are not quite on top of the front, but they're close to it. Compared to our gallopers, where their hind legs are actually landing in front of where their hind legs are landing in front of where the front legs took off. So we'll see the hind feet before we see the front feet. So something to keep in mind is wildlife operate by making decisions based on energy principles and trying to conserve as much energy as possible. So they're independent of humans for acquiring food, right? So their choices, such as the gait, what kind of food they're going to pursue, the amount of time they're going to spend in an area, their activity, the timing of activity, how often they'll be active, how far they'll travel, things like that are all based around conserving energy and making decisions on whether it's worth the energy to gain new resources. So they're trying to make these decisions and be as efficient as possible. So the reason I bring this up as I'm talking about tracks is as we're seeing canid species, so with the claw marks and with that one lobe, so we see canid species and they're in a meandering pattern. We tend to see that more with our domesticated dogs rather than our wild canid species. So our coyotes and wolves tend to have straighter paths rather than our dogs are making more meandering paths. Okay, so now moving on to scat. I admit I am not the best when it comes to identifying species by scat. Some people are amazing with this skill, but I did find a decent key which can help you step through identifying your species. And what I recommend is testing out this key, starting with species that you're familiar with, or if you see evidence of scat being dropped on the ground and you see that species and you're confident about that species, then look at the details of the scat. So you'll want to look at length, width, the shape, the color of the scat, how many are in a group. Is it one long trail or is it 10 or more in a small pile? Things like that. Location is important too. Okay, so before I change gears to our herps, do we have any questions I should address?
We did have one question from Julie, and she asked, I think it was on the first slide that you had about tracks. She's asking what animal made the snow track with the swirls and circles.
I believe that was a bobcat. I remember it being a feline species, so a wild felid. I want to say bobcat. I'll have to go back more closely and look at the measurements, but I believe that was a bobcat.
Oh, and we also had one question a little further up that asked, how would you capture a flying squirrel on a camera?
Oh, flying squirrels are so difficult, and I totally cheated. So I set my cameras right at Woodend's feeding station, and I knew the days that the volunteers were going to feed, and I played around with the trigger speeds of the camera. So I found if I had either a continuous video or if I set it to something crazy high like 10 pictures per second, then I was able to get some quality pictures of the flying squirrels. Um, so really, it was only because it was not random, accidental at all. I deliberately set those cameras where I knew there'd be flying squirrels. Now, one exciting point was the volunteers that year had seen only one flying squirrel pretty commonly. But by setting up the camera traps, I actually saw 3 on the tree at one time. So that was really exciting. So we were worried that the numbers have been coming down over the past few years, but it could be that they're spacing out the timing of when they're coming to the feeders so that only one had been seen at a time. But I did get some really good pictures. And I thought I put it on Facebook, but I don't remember if I put it on Woodend's Facebook page or if I put it on Audubon Naturalist Society. So I'll have to remember to find those pictures and make sure to put it on, share it with someone who has social media with you guys, because those were some really fun pictures.
Yeah, that's really cool.
Otherwise, it's really hard to know about flying squirrels. I mean, you really have to just have a spotlight on the tree at just the right time. They're so hard. Sometimes you'll see shadows, and you won't see what the animal was. You'll just see a shadow, and you're like, why is there a shadow at night? So all I can say is there's a good chance you have a flying squirrel. We do have them. And most people don't know about it because most of us are inside by the evening. So they're really, really hard.
We had one last question about the scat key. So they asked if like what the scat key is that you recommend. Is it just the DNR one or
So the scat key, so that one wasn't Maryland DNR. I think it was like, was it Wisconsin? Um, Maryland has referred me in the past to this little guide that you have to purchase. So it's like Tracks and Scats of the Northeast, I think it's called. But I was trying to find some resources that would be free for everyone today. So that's why I didn't mention that book. But there is a good book that we can purchase. It's still I don't necessarily think my brain doesn't work in textbook in the order the textbooks are laid out. I really like flow charts and dichotomous keys, so that's why I like the one that I posted, because it really leads you down a path of if this, then go here. If not, then go here. So I kind of like those dichotomous keys. So I do like it. I just add a word of caution. It's not from Maryland. It's out of state. So there are some species you'll see on there that we're not going to see in Maryland.
Yeah, it would actually be great, I think, for everybody if you could just share all of those keys that you mentioned before with the group afterwards. I feel like we can probably send those out.
Yeah, and I embedded them in the PowerPoints as well. So as everyone so after we send out the PowerPoint, as you click on it, it should take you to those websites, so you can have the resources free.
Perfect. All right.
Great. Okay, so now we're going to change gears from mammals over to herps. So when I say herps, I'm talking about herpetofauna, so our amphibians and reptiles. So we're going to look in a few different places for our herps. We're going to look under rocks and logs, so the areas they are naturally. We're going to look in the leaf litter. We're going to look in tree crevices, but we can create some artificial habitat. So we can create cover boards, and we can also listen for the ones that call. So frogs call at night. So we will start with some of our salamanders and looking over under logs. So whether we're looking in a terrestrial environment, we may find a certain subset of our herps, or if we go over to our aquatic environments, we're going to find another set of herps. Either way, we want to be looking under rocks and logs for our herps. So the idea is that we're turning them over very slowly. We want to roll rocks and logs towards us. Instead of away from us, we want to roll it towards us, so that we, these humongous monster predators, we are the last thing that the animals are seeing. We want to try to minimize that flight or fight response. So we want to turn it towards ourselves, and we want to be very slow. We want to be very quiet, and we don't want to jerk, make any sudden movements. We don't want to startle them. If we're in an aquatic environment, we want to moisten our hands with the water that's right there, so that we don't remove any slime coating. So we want to make sure our hands are wet when we're working in an aquatic environment. So after we've rolled over lots and lots of rocks and logs, we'll look for any critters, and we'll want to look really quickly because they are going to move. They are going to dart to try to find cover. So we're going to try to remember as quickly as we can, as much detail as we can. Then we're going to slowly put it back in position, into the same position it already was. All right. We also can shake out leaf litter. So they're also attracted to large amounts of leaf litter. So we can shake out that leaf litter, and if we shake it into, say, a clear plastic bag or net, then we also have a longer look at our herps. So I'm going to try to play this video to see if we can have a demonstration.
Shannon, was
Yes.
your intent for us to hear?
Oh, can you not hear very well?
No.
Okay, that is the loudest that video goes. So I'll just wait for everyone to watch that on their own computer when they can turn the volume up. But basically it's a demonstration of shaking out the leaf litter, so shaking it from above, so that any animal, any herps would fall down into your collection, whether you're collecting in a Ziploc bag or a net. So anything would shake in there. And there you'll also collect debris and leaves, so you can sort those out and then look to see what's left behind. And it gives you a few seconds, a few more extra seconds to look at the detail of any herps you caught. Another caution is if you are in an aquatic area, you may want to add a little bit of that water to keep your herps moist if you're in an aquatic environment, because we want their skin to stay nice and moist. We also want to look in tree crevices. So many times we'll find herps near the openings of tree crevices. I mentioned earlier, we can create these cover boards or these artificial habitats. We can lay down boards like plywood. We can lay down shingles. We can alternate them. So overlap parts of the shingles to create a gradient of temperature and moisture. We can lay down corrugated tin. So the idea is that we're creating this warm, moist environment that mimics their natural environment. And the same concept applies when we're lifting and replacing the habitat. We want to do so very quietly, very slowly, so we don't startle our animals. In this case, we won't be necessarily handling the animal. We're not going to have a plastic bag that we can funnel them into. So we're just going to have kind of that one shot to see what we have underneath it and then putting it back down. Rob, do we have any questions?
We just got one question from Eli, who said that he's normally able to find redback salamanders, but this year he hasn't found any. And he was asking kind of a general question in the chat if others have also noticed that they've been absent.
That's interesting. I saw eastern redback salamanders early in the year. I actually started looking around March. And I saw them in both color phases. And so I saw them early. So I don't know if they are changing. Now, then again, I live in Virginia, not in Maryland, but I still saw them in their normal habitat, I would expect. Now, so I don't know if they're changing the timing of their activity due to this unusual climate that we've had this past year, or could it be due to avoiding extra activity? So as you may have noticed, we are in different environments since March. So our activities have changed, and we may have influenced the location where our smaller animals may typically prefer. We may have influenced where they're located or when they're located, so they may be responding to avoid us. So unfortunately, I can't say with any expertise why you're seeing a change necessarily. I can only guess as to why you're seeing a change. But I can say the only change I saw was earlier. I saw activity earlier in the year rather than later in the year. All right. Okay, so I mentioned frog calls. So we can passively listen at night for frogs calling. I recommend moving around your aquatic environment. So so waiting until sunset, and then only surveying for a couple hours, and alternating between silence for when you're listening and when you're giving them a break. So as I approach a new part of the environment, I approach my area, I stop, I start my record keeping of my latitude, longitude timing, and then I wait a little bit for the animals to adjust to my presence, my sounds, my movements. I wait a little while because they naturally have this pause between calls, but I want to give them more opportunity to adjust. Then I start my survey for 5 minutes, and then I go on to another area. So I give them that kind of that break in time before I start my survey. But I don't stay in the same spot all night long. I'm moving throughout the area because I may hear different species coming from different parts of the environment. Okay, so now switching over to birds. So I'm sure many of you have taken many classes on identifying birds, and you know that we can identify them through their calls, through certain characteristics of how they look, through their behaviors. So what we can do for BioBlitzes is we can attract them to our area, or we can rely on them to be in the area normally. And I'll talk about some different ways to attract them. So as you've taken some classes or have read some books, you may have memorized some little mnemonic devices to help ID them, which is great. But when there's 300 species migrating through in spring or fall, it's really hard to remember mnemonic for every one of those species. So we tend to add the data we get from their call also to how they look as we're identifying their species. And so when we're looking at the bird, we're looking at different physical features such as, do they have an eye ring? Do they have a wing bar? What's the shape and color of their beak? Things like that. So some things you may not have known already. So feathers are protected, and we're going to find feathers in our backyard naturally. So whether if birds are getting in a fight, or if it's a molting period, they're going to be losing their feathers naturally. Now, you can't go and take feathers. Feathers are protected. You can leave the feather where it is, but you can take pictures, you can document it, you can measure it, and then you can go on to U.S. Fish and Wildlife Service's Feather Atlas. So this is a really fun, free tool that anyone can use that steps you through identifying your bird based on their feather. So when you go onto their website, you click on Identify My Feather, you select the pattern, you select the color scheme, you tell it the size you saw of the feather, what was the position of the feather, does it look like a tail feather, or a primary or a secondary feather. You use all those details to help key out what species that could have been. And then their atlas will show you lots of possible representatives. So you can then confidently make your ID based on this feather. And your picture of the feather you saw, you can then upload to iNaturalist as well. So for behavior, we also look for other cues. So are they flying in flocks, like these Canada geese? Or are they very gregarious, such as European starlings? Or are they flying in pairs or solitary? We can look at behavior cues, like this American redstart fans its tail feathers as it's trying to flush and forage for its prey. So we're looking at all these different cues to help us ID our birds. But do we want to rely on those cues, or do we want to attract them to our area? So many of us may feed birds to attract them to our area, but did you know that certain species have certain diet preferences? And each of these varieties of bird seed are going to attract certain species of birds. So we're already biasing our area for certain species, depending on the bird seed that we select. And also the style we select. Is it loose seed that we're putting out? Or is it the molded cake that we're putting out? Or sugar water that we're putting out. So if we put out a variety of bird seeds and a variety of styles, if you will, of bird seed, we can attract more and more species to our area. And we can also provide bird bath. So bird baths can also attract even more species to our area. One word of caution, though, is that water, you don't want that water to sit still for 24 to 48 hours. You want to either turn over your water by changing it out every day or so, because you don't want to attract unwanted insects or grow algae. So you can either turn it over, or you can circulate it with a pump, or you can add mosquito dunks or mosquito bits. And that leads us into our insects and arachnids. But before we get to surveying those, any questions we should stop for, Rob?
Yeah, we have two questions in the chat. One from Jen, who asks, why are all feathers protected? She understands that there are certain feathers, but why, like robin feathers or more common birds, why are those protected as well?
Well, I mean, until we know which species it is, because certainly we have certain species that are very highly protected. Some may require permits to gather parts of the bird or the entire bird. So it depends on what species it is. But as for why, I can't answer that at the moment. As for why the feathers of different bird species are protected, I'd have to do a little bit more investigation before I answer that. But I will say it is definitely safest to leave the feathers where we found them, and to take as much evidence as we can. So photographing, measuring, sketching, things of that nature. Take as much documentation as we can while leaving it where it is while we go back and ID. So that's the safest bet, but I have to do a little bit more investigation to see why is it that feathers are protected.
So we have someone that says, two people that are saying either the Migratory Bird Act or Saranella guesses that it would diminish the desire of killing the bird, as it has been done in the past.
Yeah. There are many possible reasons, and yeah. Oh my gosh, I could have a whole semester worth of discussion based on belief systems and attitudes and behaviors of how we feel towards animals and how our values may change, whether we can acquire parts of animals, entire animals, or whether we have to keep our distance, how our values could change based on that. So there's a whole lot of possible reasons, but the specific answer for why U.S. Fish and Wildlife Service is saying they're protected. So why do we have? So why does migratory bird? Why do there are several different acts actually. So we have migratory birds. We've got Lacey Act. We've got lots and lots of different acts that have different parts that are related. And so the rationale behind it, to answer the specific question, would take me a while. But yes, there are definitely many acts involved that we have to follow. So it's just safest to leave the feathers where we find them.
And then one last question. This one is unrelated to birds. It's actually about snakes from Dallas. And they're asking, they said they've found 3 shed snake skins in the past couple weeks and have never seen any other snakes other than garter snakes. But they think that these skins are a little bit bigger. They were wondering if you have any ideas for seeing the snakes / IDing the snakes.
Oh my gosh, just based on the skin that they're shedding. Oh my gosh, I am not good at all with IDing them based on what the skin they shed. I'm really not good. I will look into that though. So far I haven't found a key, but I will ask my herp friends if they know a key or if they're working on developing a key, because that is a really cool idea. But otherwise, it's so hard to tell what species it is, just based on the skin they shed. It can be really hard. And in our area, we do have several different species, and some that are pretty similar to each other. And it may take, looking at the color underneath to help distinguish one species from another. Habitat may help as well, but if you have multiple habitats in your property, then it can get kind of tricky because you may have several that look similar to each other. So seeing them live is always the easiest way to see all the clues to help us ID the species. So I'll definitely look into to a key. That's a really cool idea. All right, so let's switch gears over to our insects and arthropods. Okay, so I do want to take a moment to acknowledge the amazing work that Audubon Naturalist Society has done for invertebrates. Historically, we've sampled these using invasive techniques, invasive and permanent techniques. So we've used light traps, scent traps, sticky traps, water bowl traps, and the method they use is collecting them permanently, and then we identify them later. But so Audubon's, yourself, Rob, Allison Pierce, Master Naturalist trainees Christiana Fogg, Betsy Mendelson, and Bethany Draymond have developed an innovative survey technique involving the observation of the invertebrates in their natural ecosystem, a swiftly using sweep net to collect them, followed by cooling them to slow down that movement, identifying them, then releasing them back to the ecosystem. So this is a very innovative technique, as most of us have just done these permanent collection techniques in the past. So I really applaud Audubon Naturalist Society for developing this technique. Because if we're out there doing these bioblitzes, you know, if we have dozens or hundreds of us doing bioblitzes, and if we're all collecting them permanently, that's gonna have a major impact on our ecosystem. And these insects and arthropods are already undervalued in our society. I mean, just look at how many neighbors do we already have that indiscriminately spray against flying anything. So yes, I understand we may not enjoy having mosquitoes, certain kinds of bees, maybe not everyone appreciates them, but indiscriminately spraying against them is so harmful for our ecosystem. And also permanently collecting them. If many of us permanently collect them, that's also harmful to the ecosystem. So I really encourage this non-invasive technique. So another question is, do you want your BioBlitz to include all living species? So that means also trees, vines, shrubs, bushes, flowers. So the benefit of IDing our vegetation is they don't move or they very rarely move. And if they do, usually it's because we have transplanted them, but it was very intentional. So we can kind of get away with surveying for our vegetation occasionally during an occasional BioBlitz. We don't always have to do it during every BioBlitz. But it certainly could change. So the number of species of our vegetation certainly could change if we've planted something or if we've removed something. But for the most part, our vegetation species are remaining the same. So along those lines, we also have our yummy fungus that we can survey, right? So similar to vegetation, it's not really moving like our animals move, but remember that they are present or absent based on season, so based on climate. So depending on when you time your BioBlitz, you may be including fungus in your species inventory, or you may not be. So that's also a question that will help you answer when you want to conduct your BioBlitz, how many days, how many times per year, things like that. All right, so some tips. Oh, before I start, do we have any questions?
Yes, we have two questions. One related to the mushrooms is what causes a mushroom ring?
Oh, not even gonna touch that one. I'm gonna let the ID guides and let them educate you for fungus. I'm hoping to broaden everyone's minds to incorporate lots and lots of living species, not just the ones our society tends to focus on. So not just the cute, cuddly number of species, but thinking outside of that and thinking of all of our living species. So just remember to incorporate fungus, trees, shrubs, vines, flowers. Remember to incorporate insects, arthropods, and use the field guides to help you. So with that, I'm going to try to quickly go through these tips because I know we're almost out of time. So this paper, real quick, was recommending to do more than one method when surveying your species richness or your number of unique species. So in this example, they used live trapping versus analyzing scats versus analyzing owl pellets. And each method discovered a different number of species. Altogether, the species richness of small mammals was 19. But individually, one survey type led to a range of 9 species up to 17 species. But using all the methods together helped them learn there was a total of 17 species. Also looking at the relationship between the area we survey and the amount of time we survey. So increasing area, increasing the time that we survey can increase the number of species that we're seeing. Using more observers can increase. So not only do we have another pair of eyes that may have seen something that we didn't, the other observers may have experiences that complement your own, so they may be able to identify other species that you couldn't. So going back to that fungus example, maybe include an observer who does have a lot of experience identifying fungus. Oh, also safety, certainly safety numbers. Field guides, there's tons of field guides out there. Each are laid out in a unique way, and you will find the one that makes the most sense for you. And so there's usually tons to pick from for every taxa. So you'll want to make sure you have field guides. Now these do cost money, and I'm trying to give you affordable methods. So I also looked up, besides the field guides, I also looked up some apps. A lot of these are free. No app is perfect. Even if it says you can download a recording and it'll tell you the species, you know, it's never going to be perfect. You still want to go back and double check your data against what you know and what you've learned to see, was that species ID from the app correct? Or do you believe it was something else? And then when you upload it to iNaturalist, you can have someone, you can have a professional double check it. So the stages, so it's basically 4 stages of planning. So thinking out these questions, conducting your BioBlitz, posting your results, and then following through with your team members. And planning, you want to think about, you know, the scope, the different scales. You want to make your decision. You want to create your team. Will it be yourself? Will it be a small army of volunteers? If so, what are some dates and times that work for everyone? Are you picking an area that will require a permit? Think of a place to meet, to review the equipment that everyone already has, or the apps or the field guides that everyone has. So then you can make your decision if you want to purchase anything else, or if you need to borrow something from someone else. Remind everyone in advance, create an account on iNaturalist and create a project. Share that with your team members so they can upload their results as well. Here's an example of a BioBlitz data sheet. The idea is it has everyone's names, their iNaturalist usernames, a description of your site, the different methods you're using, the total number of species that you saw. You can try to include the number of individuals, but really once you get to one species, you're done. Your goal here is just finding the number of species present. If you want to know the species diversity, or the if you want to weight the evenness of the spread of all the species, that's a different research question. The BioBlitz is just looking at the number of unique species. So you want to record date and time you observed, your location, so latitude and longitude, the common name. Later you can get to the scientific name. Your evidence, somehow you want to back this up with photos or videos, things like that. Any comments. For iNaturalist, you can find videos teaching you how to set up an account, how to create a project, and then you want to share that with all your volunteers. When you're conducting your BioBlitz, you want to take really good notes, really good sketches, of as much detail as you can. You can always go back to your sketches later. You only get that one shot looking at that animal right then and there. So you want to have as much time focused on observing as you can. And then later you can go back and finish IDing. You want to take as many close-up pictures in high resolution as you can, but you want to balance that with not disturbing them. So it's kind of a tricky balance sometimes. I like taking video because it includes the visual plus audio. You can get your latitude longitude from your cell phone. Try to ID as close to species, but if you need to back up a little higher than species, you certainly can. It's fun to make a species counterboard, so it keeps everyone pumped to work, maybe 1 more hour, maybe 2 more hours, and maybe each hour you find several more species. Always be as safe as possible, whether we're talking COVID time or not. You always want to be safe, because remember, we are dealing with wildlife, and wildlife do transmit diseases. So we do want to be careful what we're breathing, what we're touching. We also want to think about preparing for emergencies. Are you in an area that has cell phone service? Or do you have a way to communicate with others? Maybe take walkie-talkies if cell phone doesn't work in that area. Bring a first aid kit. Certainly right now, we need to think about COVID, so we do need to think about social distancing and washing our hands before touching our faces or eating or drinking. Tick check, super important. So in the field, but also the next 24 hours, very important, because many times when we first see them, they're in that nymph stage where they're really small, hard to detect. So we may not see them or feel them immediately. It may be a day later. So you really want to check yourself well from head to toe, because we are in an area with lots of ticks, deer, small-footed mice. We've got lots of Lyme disease in the area. So we want to try to prevent our chances of getting Lyme disease and other tick-borne diseases. I recommend posting your findings in the field, or at least within 24 hours while the data is fresh in your mind. And you can always ask for professional grade ID. Focus on the quality of your findings, not the quantity. So it's not a race to see which volunteer can post the most number of species. You don't want to post 100 European starlings. It's more helpful if you saw European starling and an eastern bluebird. You want to make sure to thank everyone involved. You want to update everyone with your results. It's great to host a follow-up meeting to see what recommendations your volunteers may have, what may have gone better, or what could you improve on during next time, and when do you want to do your next one? Do you want it to be the next season, or do you want to wait a full year and compare your species richness year over year, or do you want to compare season versus season? So it starts with the basic understanding of biology, and as we repeat our BioBlitzes over and over, and as we're posting with others, as we're sharing our data with others, we can start to see trends in the data. Then we can make these management recommendations to improve the diversity in our ecosystems, therefore the functioning of our ecosystems. And really a healthy ecosystem is our ultimate goal. And BioBlitzing happens to be a really fun way to get there. So that is the end of my presentation. I'm sorry we ran over a little bit. We had some really great questions. And I still want to send some more resources to everyone shortly. But I appreciate everyone staying tuned a little bit longer and asking all these great questions. And for whomever wants to stay longer, I'm happy to do my best to field extra questions. Saranella, did this help you with thinking of new ways to find some salamanders?
Yes, it did. I think that what I really want to do now is be a little quieter and a little slower.
It's so easy to get excited and agitate the area. It's so easy to. And really, this ties well into future Audubon talks, and also some past ones and some classes that have been offered. So those who want to explore more detail about identifying the species can take multiple classes through Audubon Naturalist Society and through other institutions as well.
All right. We have a question from Ellie Putnam. Are there good techniques for monitoring natural sounds such as coyote howls?
So Wildlife Acoustics does make this recorder that can be programmed to record sound in a certain spectrum of frequency, and it can convert the wave files to a computer program so you can see the frequency of sound over a time period of sound. And that can help us identify certain species. It helps us identify certain species of bats, certain species of frogs and birds. Canid species have such variety in their repertoire of calls that it would be, I anticipate it being very difficult to just use a sound recording or use a sound recording and converting it to a spectrogram. I could see that being very difficult to identify canid species just because of their huge repertoire. So there's no one typical coyote call or one typical red fox. So they have so many different calls. And some of that may be behavior-based, situational-based, social communication-based. So it's very difficult to say with accuracy that one was eastern coyote or western coyote, or because where we are in DC, so we have our interesting coyotes where we have some gray wolf DNA from the north, we have some red wolf DNA from the south, and coyote and wild canids all mixed together to make our local DC area coyotes. So I would say I wouldn't rely on sound recordings alone to identify our canid species. I would like to look physically at them, whether it's live or using cameras. I'd also like to look at their tracks. Scat would be great as well, but certain studies I've read with scat, based on even the most experienced biologists, still get the scat ID wrong between canid species. They're just so similar, and depending on the season, their diet may overlap. So we can't completely rely just on the scat or just in the tracks. So it's helpful to have multiple methods to be able to ID them. So I wouldn't put too much stock into the sound recording for canid species. I hope that helped.
Thank you so much.
Great. Thank you, everyone. And you all assigned me some homework as well, so I'll get started on that soon.
Thank you, everyone. Have a good night.
Thank you.
Thanks, Shannon.
Yeah, absolutely.
Thank you.